Now we have our introduction, and so we're going to move on to with our other speakers. Tom Anderson is from the National Oceanography Center. He is going to talk about stoichiometry of mesopelagic zooplankton and carbon sequestration in the ocean. So thank you, Tom. We're ready.
Well, thank you, Debbie, and thank you for Mike for inviting me. It's a lot of years since I've been to Woods Hole, and I have to say it's an immense pleasure to be back here amongst such a great community of people. So I'm going to be talking today about stoichiometry of the ocean, but actually particularly detrital systems. And although I'm mostly focusing on the ocean, a little bit of what I do will, or a lot of what I'm talking about will nevertheless apply to freshwater systems, sedimentary systems. So you see, it's all relevant. So there's the structure of my talk. I'm going to give you a basic introduction to zooplankton stoichiometry. I just want to emphasize a bit about what optimal nutrition is before we start. It's such a critical concept. And then I'm going to move on to kind of the meat of my talk, which is about ocean carbon sequestration. I want to show some recent work that I've done on mesopelagic zooplankton, microbial gardening, and some conclusions at the end. So starting off, here's my picture of a copepod. So stoichiometry in the case of copepods is all about, we've got two currencies here, carbon and nitrogen. If you eat food with very carbon to nitrogen ratio, the organism has a ratio, a homeostatic ratio, for example, in its body. How much will go into the body, how much will go into fecal pellets, how much will be respired as carbon, how much will be excreted as nitrogen. And where all that carbon and nitrogen goes matters for how food webs and ecosystems work. If it goes into fecal pellets in the ocean, for example, it'll sink. If it's respired, it won't. It'll be remineralized in the surface ocean. So all this matters for how food webs work. So just my little bit of introduction then to zooplankton. A typical C to N ratio, molar ratio in zooplankton, that's krill, is 5.5. So what does this mean for optimal nutrition? And one thing that at least some people have kind of got caught up with is thinking, well, the optimum ratio is 5.5. It's not. So the animal will, in theory, require, at least for growth, a ratio of 5.5. Now I've split carbon and nitrogen. Carbon and nitrogen, of course, don't cycle as elements. They cycle as biochemicals. So I've normally put carbon and nitrogen together there in a protein molecule and an extra carbon in lipids and carbohydrates to make up the difference to 5.5. So as well as growth, organisms have maintenance requirements. And I've started off here with a requirement which is exactly the same. Organisms have protein turnover or biomass turnover, and all that turnover has to be replaced. So that will also be in a ratio of 5.5. But organisms require extra carbon for energy, which you primarily get, or ideally get, from carbohydrates and lipids. So that means that the ideal C to N ratio of this organism is it needs 5.5 for this, and extra carbon. So the optimum ratio is greater than 5.5. Maintenance also has nitrogen in it, which is here. But overall, maintenance has a high C to N ratio. I'm focusing on nitrogen. The same things can apply to other elements, such as phosphorus. And if you really want to get at then optimal nutrition, you need to think a bit more than that. You've got your biomass, you've got your maintenance. There are lots of other things to think about. Stability. If you eat celery to get your carbon, it's very different from eating donuts to get your carbon. You've got to do, it takes a lot to digest the celery. So the form of the carbon, whether it's something that's nice and nutritious or whether it's easily digested, the correct word is absorbed, matters. Of course, it's not just macronutrients like nitrogen, phosphorus, but micronutrients like fatty acids and amino acids can be limiting. And indeed, that's going to be the topic of my talk today when I get to detrital systems. And food quantity matters. So food quantity affects the relative availability of food for maintenance versus biomass. Basically, you have to meet maintenance first. Once you've met your costs for maintenance, then you can start accumulating new biomass growing. Some things are temperature dependent. So temperature dependence is actually a very topical subject at the moment with global warming, but particularly food quantity and maintenance. And putting it all together, you can see you can take the C/N ratio of an animal, its food, and because those ratios are different does not necessarily mean the food is imbalanced. Because I've already said that you need, in principle, a higher C/N, at least the scheme I showed. Put all this together and you get what's called a threshold or elemental ratio. That is the optimum ratio in food, and if you're either side of it, it's a threshold. You're either carbon limited or nitrogen limited in this case, depending on what currencies you are. And any excesses, carbon and nitrogen, have to be disposed of, either respired, excreted, put into fecal pellets, gotten rid of somehow. Okay, that's my basic introduction then to zooplankton stoichiometry, and a little bit introduction to carbon sequestration in the ocean. You're probably mostly familiar with this, the famous Martin curve. So there's a series of experiments done off the west coast of the USA and measured particle flux with depth, and you can see the different sediment traps, and here are the different zones. This is the mesopelagic zone, nominally between 100 and 1,000 meters, which I'll focus on today, and you can see the flux declines with depth, and this is, for the mathematicians, the modelers amongst you who do this kind of stuff, that is a kind of ingrained equation which describes that curve. The reality of what's causing that curve, there's a lot going on, and that's where the action really is at the moment in terms of export flux in the ocean. This study came out 3 years ago now. This is a mesopelagic zone at a station in the North Atlantic. These are the inputs as measured, and these are the outputs. That's input to the mesopelagic zone. These are respiration fluxes by zooplankton and microbes. And you can see that the microbes dominate respiration. And I actually put this model together, and it suggests that actually the microbes and the zooplankton each utilize about 50% of the flux coming in. This is detritus flux. But the zooplankton mostly break it up and pass it on, if you like, to the microbial food web, which mostly respires it. So there's this dichotomy that zooplankton, while they're utilizing the detritus, they're actually not actually eating, if you like, a lot of it. They're just breaking it up. And that subsequently fuels the microbial food webs. This is really important. We have to understand this if we're to get at carbon flux in the ocean. Because whether a particle is used by bacteria, which will remineralize it straight usually back to CO2 and nutrients, or whether it's used by zooplankton, which might produce fast-sinking fecal pellets, matters. And yet we have a relatively rudimentary understanding about this balance between zooplankton and bacteria as yet. So I'm going to be talking about the zooplankton side of things today, but it relates to the microbes. So here's Debbie Steinberg's famous fecal photo. There's some nice poo in the ocean, and of course size matters. There's salps, copepods, and euphorsids. And it doesn't look very nutritious, does it? Wouldn't like to eat that. You only have to look at that to think, yeah, that's not great for zooplankton to eat. But what you can't see on that picture is detritus is colonized by microbes. So you have these are flagellates, you have bacteria, and then bacteria fed on by the flagellates. You have this microbial community living on the detritus. And while this is crap, literally, this is nutritious. This stuff is nutritious. It's full of nice, the microbes synthesize fatty acids, amino acids. They're rich in nutrients, but they're very low in biomass. But nevertheless, there is an opportunity there for the zooplankton to get at all that new. It might be a small in biomass, but actually it might be a really important part of the nutrition of the zooplankton. This is where the action, at least to me, really is with this detrital stoichiometry at the moment. Though it's been knocking around a while, and as I said, there are links to other systems. These are freshwater shredding insects. I don't know much about them, but I think that they eat decaying leaves in streams. So you have these decaying leaves, and again, this is a bit akin to the detritus in the ocean. It's not very, you can just look at it, you can see it's not very nutritious looking, but growing on those leaves are fungi. And the fungi are, again, rich in micronutrients and low in biomass. And this is all put forward as the so-called peanut butter cracker hypothesis that I learned about. Actually, I was talking to someone from Freshwater side yesterday, or 2 days ago at my post, and they said, oh yeah, peanut butter hypothesis, well known. It dates back to the 1970s. The oceanographers have never heard of it, apart from me. So basically, that's your cracker. But actually, in the ocean, I think it's more like cardboard, or I wouldn't like to eat it at all. And that's your peanut butter. And again, the difference is there's quite a lot of peanut butter on this cracker, but in the ocean, I think it's more like a speck of it, a very small amount. But there's the analogy. And so what I'm going to be talking about, and what a study I've done recently, is to try and look at to what extent can the nutrition, in this case of shredding insects, but of copepods in the ocean, depend on that peanut butter as opposed to the cracker. And just to make the point, the difference in biomass is enormous. This is typically, I think, 1% biomass. The biomass of microbes is this relative to this. So you've got a choice of an enormous cracker or a bit of cardboard that is pretty nasty stuff, really, versus a very tiny amount of nutritious stuff. And as I say, it really matters. A question relating to all of this is, do the zooplankton even eat this, as in the sense of ingest it? They process it for sure. They are actually capable, I'm told, I've not seen it, of literally scraping the microbes off the detritus. Again, we really don't know enough about all of this. So I'm going to at least try and present a case that this can be, at least theoretically, a major part of the nutrition of these detritivores. So we move on to my third part of the talk, which is mesopelagic zooplankton. So the mesopelagic zone, as I said, is 100 to 1,000 meters in the ocean. It's where all this important turnover of detritus occurs, a stoichiometric analysis. And this is work I've recently published, and I want to acknowledge my two co-authors here. They're both great guys, David Pond, Dan Mayer. So we've recently published that for anyone who wants to look at it. And this is the model we use. It's a very simple steady-state analysis, where you just start off with a certain amount of detritus. And the detritus can follow one of two pathways. The zooplankton here, copepod, if you like, can eat it directly, or it can go, the detritus can be used first by bacteria, and then the bacteria are grazed on by these flagellates or protistan bacteriivores. And so as you go this way, of course, there's at each step there's a large loss of biomass because of the bacteria have low growth efficiencies, the low growth efficiencies involved, and then primarily these, in fact, are eaten by the zooplankton. So the zooplankton have these two sources of nutrition, directly on that, which is non-living, or these living components at a much lower biomass. And I'm not using carbon and nitrogen as currencies in this model. I've got carbon and essential fatty acid, docosahexoenoic acid. It's a bit of a mouthful, it's DHA. And this fatty acid, we need it, animals need it, it's used particularly in membranes, in reproduction. Zooplankton, for the most part, cannot synthesize it. Phytoplankton microbes can. And we, for example, eat it in fish. We buy it in tablets, what have you. So it's very important fatty acid. One of the so-called essential fatty acids because it cannot be synthesized by the zooplankton. So coming back to the model, just to complete it, we've got the whole model is in carbon. So all the flows are specified by carbon. Basically, these are just simple parameters where you specify a fraction of carbon going one way or the other. So they're simple growth efficiencies and transfer efficiencies. There are no fancy equations in this model, just a few parameters which are difficult to constrain. And the red arrows are both carbon and DHA. So you can see all the arrows going into the zooplankton are either carbon, have both carbon and DHA, hence we can analyze the stoichiometry, the nutrition of those zooplankton in those two currencies. So if you look at a bit more about the stoichiometry, this stuff, there's lots of it. So it's high in carbon, but it's low in DHA. So the animal that first produced is the herbivores, if you like. I'm talking now about detritivores, but the herbivore or the animal that produced this in the first place stripped out all the nice stuff and just left the nasty carbon and not much in the way of DHA. So anyway, these are values we got from the literature. It's a bit difficult to get these values, but sometimes they're taken from surface water samples, or have you actually getting samples from the mesopelagic zone? Well, that's something to be done, much greater. And a great purpose, of course, of models like this is to try and inform field programs as to kind of things that might be useful to measure. In contrast, the flagellates, bacteriivores, they're high in DHA relative to C, and they have so they're relatively high DHA to C ratio, and you can see here that's Copepod, and this is also high, relatively high, so much higher. So you can see this in DHA, so you can see this is a poor substrate, this is a relatively good substrate, but there's not much of it. It's not just about the ratios, as I said, it's about the absorption efficiencies. So we've got here this stuff. So absorption is the same as digestion. Absorption is the correct word, if you like, for digestion. So this has a low absorption efficiency, just 10%, and indeed, my co-authors, particularly Dan Mayer, says, well, even that's very generous, and it ought to be a lot lower than that. You could be talking about going down to 1%, 10%. And it's not just the carbon at the DHA. We assume that they can't selectively strip out the DHA. Wouldn't be that easy. Whereas these have a high absorption efficiency, so they can be readily digested, like 72%. And the other key parameter, there are various parameters, but the other key parameter is the bacterial growth efficiency. So bacterial growth efficiencies in the ocean are low. And a typical value that you can get is plus or minus a bit, but 0.12, 12%. So that means that if this stuff, which is bacteria use this, so bacteria do grow on this, and they can synthesize the essential micronutrients. But when they do use this, there's a big loss, an 88% loss of carbon from here to here when you're producing the biomass. And then that's just a bacteria, and then when you go from bacteria to flagellates, there's another trophic loss. So what I'm going to do is describe two pathways on the next graph. Blue is just, I'm going to look at the two theoretical ends of the spectrum, if zooplankton eat just detritus or if zooplankton eat just these to start off with. See, just to compare them, and I've got blue and orange on the next graph. So I'll take you slowly through this graph because this is the key graph for the analysis. This is ingested carbon and DHA. Blue is the detritivorous pathway, detritus only. Orange is the microbial pathway, microbes only. Everything is normalized to 1 for this blue column here, 1 unit of carbon, detritus. So the detritus is relatively poor in DHA, so that bar is less than that bar. The microbial pathway, if they're just eating microbes, you can see that the amount of carbon is real low due to those trophic transfer efficiency losses, and it's relatively rich in DHA. That column is relatively high relative to that one, but still real low. If you then go to absorbed, which is the next graph here, absorbed carbon and DHA. There's a 10% absorption efficiency for the detritivorous pathway. So these two bars are exactly the same as these two bars in proportion, but you can see we've gone from 1 to 0.1. When it comes to the microbial pathway, these bars move up because these carbon and DHA in the microbes are absorbed with a high efficiency, 72%. So these bars kind of suddenly shoot up. And by comparing these bars here now, you can work out which, based on the heights of those bars, what the growth will be or what will be limiting. If you take the detritivorous pathway, you can see there's still less DHA, and the detritivorous pathway is limited by DHA if they were eating detritus only. The microbial pathway is limited by carbon because that bar is lower than that bar. And the two limiting bars in the middle convert into growth here. And actually, the result there shows that it's almost equal, whichever way. It's just the way it happened to work out. We didn't tune this model in any way. It's just the way it worked out. On either end of the spectrum, it's almost equal. In fact, the detritivorous pathway wins by a fraction. Which tells you that microbes are at the very least a very strong case that microbes may be playing an important part in the nutrition here. So then we can look at, if instead of looking at the two ends of the spectrum, if you take the whole way from pure blue, which is detritus only, to microbes, and you look, which here is the other end, 100% microbial. If you allow them to eat a mixture of detritus and microbes, this is the carbon to DHA ratio of the flagellates, the microbial flagellates. You can now see there's an optimum here, and it's dominated, it's right on the microbial end, about 80% microbial. So again, if they were able to access optimal diet with the parameterization as I had it, which I say was not in any way tuned, it's strongly microbial. So again, there's a strong case that microbes are dominating, could be dominating the diet. Another very interesting point is this effectively is a growth efficiency. It's really a trophic transfer efficiency because it's a question of what they actually ingest, and it's real low. It's less than 2%. So that's saying of the detritus coming into the mesopelagic zone in this case, only 2% actually makes it into the copepod biomass. That's a huge loss and has big implications both for the mesopelagic food web and, for example, mesopelagic fish, which is another big topic, which I don't have time to talk about today, but it's a really interesting one, and carbon flux via sinking fecal pellets, etc. And just allied to that, do the animals even eat the detritus? They break it up, and we'll come into that, but do they even eat it? Again, very important for carbon flux. Just to show you can do a sensitivity analysis for different ratios of carbon to DHA in the flagellates, and you get a similar result. So we have done sensitivity tests on that. Time's moving on, so I'm going to move beyond that graph and go into my last main section, which is microbial gardening. So there's a question, why are zooplankton so wasteful with this stuff? Well, I think it's to do with the microbes. I think we've reached that far. Are the sinking particles simply too big? I don't think it's as simple as that. Is sinking detritus not really their food at all? Yes, very possibly. Do they just scrape the microbes off? I mean, a lot of this is just hypothesis. A big part of what I'm saying is this model is just trying an appeal to try and get more information to firm up on some of this stuff. Microbial gardening actually comes from work done by people like Tom Fenchel way back in the 1970s. The idea is that the larger organisms, such as the zooplankton, but here we've got a sediment system, we've got worms in the sediment or zooplankton, they actually actively break up that detritus, which is where I came from much earlier with the carbon fluxes. They break it up because by creating smaller particles have a greater surface area to volume ratio, and they're more readily colonized by microbes. And so they're actually gardening microbes. And this idea comes all the way back to the 1970s. It's a bit controversial, but, well, it kind of makes sense. Here's another example. There are lots of examples from all sorts of organisms. These are leafcutter ants. So leafcutter ants are pretty destructive. They actually cut leaves off plants, cut them up, take them into burrows, store them in the burrows, but they don't eat the leaves. The leaves are just left in the burrow, and like the peanut butter on the cracker, the fungus grows on the leaves, and the ants eat the fungus, and the leaves are then put away in dump chambers. They never actually eat the leaves as such. It's just another example of gardening, microbial gardening. And the question is, does microbial gardening occur in the twilight zone of the ocean? As yet, it's only a hypothesis, and I can't answer that today, but I can put forward the case. So is the reason that copepods are fragmenting all this detritus, which is really important for export flux, it slows it up, etc., simply because they're trying to culture microbes on it? And here's another model. I won't go into great detail, but actually this is a carbon-only model. It has two types of detritus. D1 is large sinking detritus. D2 is smaller detritus that's been broken up, if you like. And you have a whole little food web here, all the little circles refer to which color the arrow is going to in the scheme of things here. But the key thing is the two types of detritus, again, being eaten by detritivores. The large detritus has low accessibility to microbes. The microbes aren't growing on it. So it has the detritivores, again, we've got a 10% absorption efficiency on it. The small detritus is used by high accessibility to microbes. The microbes grow on it. You have a high absorption efficiency for the zooplankton, and again, BG of 0.12. It's actually a similar model to the last one in many ways, and this is the result we got, but we're specifically looking at gardening here. So here we've got the amount, on the x-axis we've got fragmentation. So on the left-hand side there's no fragmentation, in which case you start off with you have large detritus sinking into the mesopelagic zone. So if there's no fragmentation, the animal just eats something which is low accessibility to microbes, something that's quite nasty, and the growth is low. As you allow fragmentation to carry on, the blue goes down, the large goes down, you get a smaller amount of small detritus building up, but at a lower rate because the microbes are degrading it. But because this is so much more nutritious, production goes up. And this kind of makes it's only a theoretical case based on best estimates we can come from for the parameters. It's a theoretical case which says this promotes microbial garden. The more you fragment, the more you garden, the more you grow. It's just a purely theoretical analysis. Again, I say we wrote this actually was published 3 years ago in Bioessays. I should particularly credit Dan Mayer for a lot of this work. Okay, so I'm just about there. Just my conclusions to finish. So conclusions quickly. Optimal diet depends on requirements for growth and maintenance. Detritivorous zooplankton may obtain nutrition from microbes associated with detritus rather than a non-living substrate. Zooplankton may fragment particles as a means of microbial gardening to promote growth of microbes. The extent to which this zooplankton ingests fragment detritus has important implications for sinking rate and carbon sequestration, and there are lots of uncertainties in the system, lots of work to be done on the physiology and ecology of the organisms concerned. That's it. Thank you very much.
Jenkins, University of Rhode Island. That was a really thought-provoking talk. And I just had a question for you about the rates of bacterial growth efficiency. Were those rates actually measured on the particles, or are these bulk water column rates for the mesopelagic?
I took particle-associated bacteria as opposed to free living ones. You have to take account of, it's a bit awkward because the particle-attached bacteria produce exoenzymes, and so you get firstly a loss due to just the ordinary loss, metabolic losses, but you also have to take account of the immense losses into the water column, and so it takes account of all of that. Yeah. But again, those numbers and the methodologies, that community always has a trouble quantifying.
Yeah.
Difficult one.
So has there work been done where it's been scaled by size of the fecal pellet and those rates?
No.
Yeah,
no.
That'd be really interesting. Yeah.
I'm not an expert on where all those numbers come from. I've read the basic publication, no, but there's relatively little information on it. Most of the work on BG, bacterial growth efficiency in the ocean, has been done on the free living bacteria that are in the water column.
Yeah.
It's tougher to do it on the particles.
Yeah. Thanks.
Thank you. I actually agree with you about the microbial gardening idea, but wouldn't it be rather altruistic behavior to break up particles that then sink away and get utilized by some other organism?
I don't have a problem with altruistic. Providing there's enough of the stuff around, then I personally don't have a problem with altruistic type hypothesis. It's exactly the same for the the particle-attached bacteria, which produce exoenzymes, which go, digest or the particles, but most of it's lost in it's your surrounding organisms. I kind of put forward, I have to say, when you say I agree with the microbial garden, great. It's good to hear that people, it's still very much a hypothesis, and I present it as such, as opposed to any kind of fact. It's something that needs to be investigated. Same question.
Anitra Thorhawk, Yale University. In the shallow water systems, there's something quite equivalent to your detrital ecosystems, wherein there are tubes made with about 10 species which break down the detritus. And my question lies in what happens to these in the catastrophic events, because frequently they fill up with imported sediment, and then all the animals and so forth just leave their stoichiometry there and go off and move. And I'm wondering, in the deeper sea, what occurs with catastrophic events to these systems you were describing.
You're asking about the deep ocean?
Well, the systems you were talking
so
about.
I'm, of course, primarily talking about epipelagic systems. That's water column systems, nothing to do with benthic or on the bottom.
How deep were your systems?
So the mesopelagic or mesopelagic zone goes from 100 to 1,000 meters. So in the open ocean, that's still a long way from the bottom. You then have the battle. Typically, the deep open ocean is several thousand meters down, so 3,000 or 4,000 meters down. So there's no influence of the bottom of sediment or anything like that. So a catastrophic event will simply will mix everything up in the ocean, and that will have a big effect, but not in the same way as destroying a sediment type system or something which has a bottom.
Thank you.
I'm over here. Debbie. Hey! Susanne Neuer, Arizona State. So I have a comment on the evolution of the altruistic behavior. So I think we really have to make a distinction. If it's a leafcutter ant that has a whole colony, many millions of individuals, it makes a lot of sense to do gardening because it benefits the entire, and there's altruistic behavior that evolved. Now, that is not known of copepods, right? That kind of colony type thing. So what I'm rather more comfortable with is that they take those particles and they eat off the peanut butter, and as a side effect, they fragment the particle because they have to hold on to it somehow. And so you've seen those videos that Sari and Morten Iverson, I think, has made them. The copepod holds on to the particle, twirls it, probably eats off it, and then as a side effect, maybe it fragments.
I like that idea. Look, I mean, the microbial gardening, I put it second. So the way I did my talk was I presented the pure stoichiometric case first, carbon versus DHA microbes versus the raw detritus. And that stoichiometry is not predicated in any way on the microbial gardening hypothesis. It's just the way it is and you don't need to have any gardening in order for that stoichiometric analysis to hold valid. And then I kind of went the extra step, and it's ironic that we did it the wrong way around in the publications. We looked at the gardening first. I then went the extra step and put forward the microbial gardening hypothesis. And so there's this idea that they culture microbes. But look, I mean, I'm kind of in agreement with you. I'm totally open-minded about just precisely what is going on there and why the zooplankton are. I'm not suggesting that necessarily the microbial garden.
Yeah, I don't mean to.
No, you're not. Your point is very well taken. We need to understand just what's going on with particle distributions in the ocean. And so why do zooplankton do this? The microbial gardening hypothesis is just a hypothesis, and you can easily come up with very similar hypotheses, such as the one you just stated, and I think we really need to look into those.
So the other point I wanted to make, you showed the different sizes of fecal pellets. I think that those big salp fecal pellets, there's they sink so quickly. I'm not sure how much they can really be used to be fragmented. Probably there's a size spectrum of fecal pellets and particles that needs to be taken into consideration.
The dominant grazer in the mesopelagic zone is going to be the copepods. I put up the fecal pellet picture with the salts on because it's such a wonderful picture. But,
we all agree.
yeah, right, so that's why I do it. But so really, I agree. The salt fecal pellets will just go straight through the system, and very little happens to them because they're sinking so fast. Okay.
Great. Thank you. I think we'll move on to the next week. We'll have some more time to ask